welcome to part 2 of biotechnology, where we're talking about PCR and gel electrophoresis, which is actually quite fun. So, first we're going to explain the purpose and steps of polymerase chain reaction, justify why a special DNA polymerase is needed for PCR, compare and contrast PCR with DNA replication in terms of enzymes used, the steps, whether primers are needed. We're also going to explain how gel electrophoresis separates DNA and interpret a DNA gel. Okay. So, polymerase chain reaction, the abbreviation here is PCR. This is used to copy a DNA of interest in 3 steps. The first step is a really hot step. So I call these, the names I use, like heat, is kind of relative, but this is seriously hot. 98 degrees Celsius is almost boiling. 100 degrees Celsius is boiling. Remember what happens to proteins at high temperatures. They denature, they lose their structure, they lose their function. So this is a very sort of interesting step, and this is why we need a special DNA polymerase that can withstand this heat. Because remember, DNA polymerases are enzymes, and therefore they are proteins. So temperature is one of the environmental conditions that affects protein function. So we need a special protein here because the first step is heat. So this step, the purpose of this step, is to separate the 2 DNA strands. In DNA replication, this step is performed by DNA helicase. But when you're doing this in a test tube, you don't want to pay for extra enzymes. And why should we pay for an extra enzyme if we could just almost boil this reaction? This doesn't take place in a living cell. It won't hurt the DNA at all. And as long as we have special enzymes that can withstand the heat, it's totally fine. The second step is the cool step. So this is 55 degrees Celsius, which is about 131 degrees Fahrenheit. So in reality, not actually cool. You don't actually want to touch this, but it's much cooler than the hot step. So heat step denatures the DNA, separates it into 2 strands. This takes the place of helicase. The cool step allows the DNA to close with the primers that tell us where, well, it will tell, not us, tells the DNA polymerase where in the genes to copy. So if you want a specific region, you make your primers for that specific region. And this cool step will stick those primers together with the DNA. The next step is the warm step. And this 72 degrees Celsius is the optimum temperature for that special DNA polymerase, the Taq polymerase, to do its function of base pairing and making that second strand of DNA. Okay. So the animation for this was posted on Blackboard, and here's the link again. It's on YouTube, right here. I suggest that you watch it again now. And be sure to take note of the enzymes used. Okay, so moral of this story is that PCR is a way to create multiple copies of a specific DNA segment. So remember, in DNA replication, that's part of mitosis. And in onion root cells, for example, that took 12 hours to get through 1 round of mitosis. And so PCR is a way for us to copy these genes whenever we want. Additionally, we can copy just small segments, just the parts that we need, or we can copy the entire genome. And here's a flow chart showing how many PCR cycles have gone and how many DNA copies we get. And we see that this is actually exponential growth. So we start with 1, in the second round we would get 2, and the third we would get 4, and then 8, and then 16. So after about 30 cycles, we have millions of copies of DNA. So we can make millions of copies in the matter of hours. So let's quickly compare PCR with DNA replication. So let's start with DNA replication because we've covered this extensively in class and in lab. DNA replication, the enzyme used, DNA polymerase, DNA helicase, DNA ligase. There are 3 enzymes that we use. The DNA separates using DNA helicase. Primers are not needed for this reaction, and if we start with 1 copy of DNA, we end with 2 copies of DNA. In polymerase chain reaction, the enzymes used our Taq DNA polymerase, and remember we do not need a helicase, and we do not need ligase. So it's just Taq DNA polymerase. This saves a lot of money. Okay. DNA separates using heat instead of DNA helicase. And this heat that separates the DNA is the reason why we need a special enzyme, because a normal DNA polymerase would completely die in that hot step, that step 1. Primers are needed here to direct DNA polymerase where to copy. In DNA replication, primers are not needed because we copy the entire genome anyway. And the number of copies in polymerase chain reaction is on the order of millions. Millions of copies from a single reaction. It's amazing. Okay, so that's all I wanted to say about PCR. So now let's cover gel electrophoresis and DNA fingerprinting. So gel electrophoresis separates molecules by using an electric charge. So that's what the electro means. And the gel part is actually made of agarose, a polymer that makes like this sort of mesh looking thing. So here's a picture of agarose, and this is what the inside of a gel would look like. So you can see it's just basically a mesh made of carbohydrate polymer. And so you can think about this, you know, like if you have ever had to like crawl through like those army ropes, it's something like that. So this is a way that we can separate molecules based on size, because the larger molecules it will be much more difficult for them to get through these tiny pores, and the small molecules will just glide right on through. Okay, so this is a picture of an agarose gel in a casting tray, and we're going to do a lab with this in just a little bit. So we see these like little rectangles. Those are from combs, which basically make little holes in this jello-like substance. Those holes are called wells, and that's where we put our sample. Then, when we apply an electric current, the sample will separate down this lane beneath each well, with the larger molecules at the top and the shorter molecules at the bottom. Okay. Carbohydrate polymer mesh, etc. Okay, so electrophoresis separates the DNA by size by applying an electric current to it. So consider the chemical structure of DNA. What is the net charge of the DNA molecule? So think about DNA. We love DNA made of nucleotides, sugar, phosphate, and a base bonded down one side. The sugar does not have a charge. The base does not have a charge. But the phosphate does have a charge. And this charge is uniform throughout all DNA molecules because phosphate is part of the backbone. So quickly look up the charge of phosphate, and then you'll be able to answer this question. So once you've found the charge of phosphate, remember that opposite charges attract. So if we're putting our sample up here, this is where our phosphate charge will be, and at the bottom here is where we want the opposite charge to be. So would that be gel A, gel B, or gel C? Because remember, if whatever charge the phosphate is, it's going to head towards its opposite charge, whether that's to the top or to the bottom. If there's a charge on both sides that's the same, then nothing will move. So you know your options here are either A or B. This will be on a quiz. Okay, so now that we know about gel electrophoresis and recombinant DNA and all of these things, we can talk about DNA fingerprinting, which is a combination of techniques that's used to identify, for one example, whether a sample from one place is genetically related to a sample from another place. So a lot of times this happens in paternity tests and in crime scenes. Okay, so here's our DNA, shown here as the blue line, and we can take restriction enzymes. Remember, restriction enzymes are sequence specific. So they're going to cut it in specific areas, and each person's DNA is their own sequence of DNA. So when we cut this, And all this happens in a tiny little vial that looks like this. Each person is going to have a different pattern of cut bands. If you're closely related, then your pattern will be more similar. Okay, so all of this happens in this tiny vial, and then we can look at the different DNA sizes using gel electrophoresis. So we've used restriction enzymes to cut up our DNA into our particular pattern, and then we put this in the gel, we load our well, and we apply our electric current. This will separate the DNA pieces with the highest molecular weight at the top and the lowest molecular weight at the bottom, because those skinny ones can wiggle through that mesh the fastest. Okay. So now we look at the pattern to determine which two are the most similar. So, lane 1 is DNA from a crime scene. So, which suspects should be detained for further questioning? So, and you're going to notice that this is not going to be a perfect match, because in real life, nothing is 100% certain, even DNA testing. We can be 99% certain, but, you know, that's such is life. So, you want to figure out which one of these, 2, 3, or 4, is the most similar to number 1. So the way I do this is I count the bands here, 1, 2, 3, 4, 5, and then I count how many match here. 1, 2, 3. This only has a 3. This has 1, 2, 3. This has 1, 2, 3, 4 matches. So that tells you that this one has the most number of matches to lane 1. And that will help you answer which suspect should be detained for further questioning. Okay. So we've already covered this. Are the shorter DNA fragments at the top or at the bottom? Explain why. So the shorter fragments will be at the bottom because they can wiggle through that mesh the fastest. So I know that this is horrible to say, but it's kind of funny and it helps you remember. But the fat DNAs can't run as fast. The skinny DNAs will run the fastest. So if this is our starting line, the skinniest ones make it to the bottom, and the fat DNAs stay at the top. Okay. So we've got shortest at the bottom, largest at the top, and here I've labeled several bands A, B, C, D. Okay, so in lane 1 here is what's called a ladder. This has known sizes of DNA. And we can use this to help interpret the sizes of similar bands. For example, if it lines up with here, we know that this is about 100 base pairs. So these bands would be 100 base pairs. If it lines up with the top one, we know that this one's 1250, so these two would also be 1250. So ask yourself, what is the size of fragment A that's right here, and then you just compare it to the ladder to figure out what size it would be. That's the trick to answering these questions, and it is never tricky. Okay. So, now we're going to apply interpreting a gel with recombinant DNA. So the vector is band B, right here. The gene of interest is band A, right here. What is the size of the recombinant DNA molecule? So given this and this, we should be able to figure out which what the size of the recombinant DNA molecule is, and also which band that would be. So this should not be tricky. The end.